Hello everyone, welcome to MMBS111 Introductory Microbiology. The next several mini lectures come from Chapter 9, where we're going to be discussing how we can control the growth of microorganisms. So this mini lecture will address concepts from Section 9.1, generally speaking about microbial control and some of the means that we can do that. There are 4 learning outcomes for this mini lecture. First, define sterilization, disinfection, decontamination, antisepsis, and degermination. Identify the organisms that are the most resistant and least resistant to control measures. Compare the action of microbicidal and microbistatic agents and provide an example of each. And finally, name 4 categories of cellular targets for physical and chemical agents. So, when controlling microorganisms, we want to limit our degree of exposure to those organisms that are potentially harmful. And that has been an ongoing concern, especially in modern medicine, trying to prevent exposure to those organisms that can cause disease or cause additional problems. There are several ways that we can control microbes outside of the body, and those are listed here on the slide. So sterilization, disinfection, decontamination, which is also known as sanitization, and antisepsis, which is also called degermination. Each one of these techniques has a different outcome as far as controlling microbes and the level of control that we're able to achieve by using one of these methods. So in the table you can see each of those 4 terms that you were introduced to. It's a basic definition of the term, and then some key points to keep in mind when we're applying this process, and then what are some examples. So sterilization is a process that destroys or removes all viable microorganisms, and that includes spores and viruses. Sterilization is generally reserved for non-living things, inanimate objects, because it would be impractical as well as dangerous to sterilize parts of the human body. And as we'll learn in later chapters, many of these microbes that colonize the human body are really important for us. Some examples of sterilization are high heat. So if you're familiar with an autoclave uses incredibly hot steam in order to sterilize tools, glassware. It's how we sterilize biomedical waste. And then there are some chemical sterilants that we can also use that are capable of destroying endospores. And there are some examples there: ethylene oxide, peracetic acid, glutaraldehyde, and some types of radiation can also be sterilants. Disinfection is a physical process or a chemical agent that is used to destroy vegetative pathogens and toxins, but is not effective against endospores. So it's a step down from sterilization. Again, it's normally used on inanimate objects because concentrations of a disinfectant that are effective are typically harmful to human tissue. So things like bleach, iodine, boiling, those are methods and agents of disinfection, and not really wise to use on live tissue. Next is decontamination or sanitization, and this is a cleansing technique that mechanically removes microorganisms to safe levels. And This is a method that's important in the food industry. This is also essentially home cleanliness. We are sanitizing. This involves the use of soaps and detergents, dishwashers, those kinds of things. Help to reduce the numbers of microbes present. And then finally, antisepsis, also degermination, reduces the number of microbes on the human skin and tissues. So this final process is really the only process that is appropriate for the human body. It involves scrubbing the skin and sometimes use of chemicals like alcohol or surgical hand scrubs, those kinds of things. So this table is from your book, and it shows you the 3 types of microbial control methods. So there are 3 ways that we can, or 3 processes that we can use to control microbes. The first is physical agents. So these don't involve the use of chemicals. It's actually a physical process. So you can see the 2 main physical agents are heat and radiation, and we're going to talk about the types there. Some of these methods result in sterilization. Some of these methods result in disinfection. And a lot of it has to do with the time of exposure as well as the temperature. The second method is the use of chemical agents, and we're going to discuss chemical agents in the final section, section 9.3. Some chemicals result in sterilization. A larger part of chemicals result in disinfection. And then, as you've seen, there are some chemicals that we can use as antiseptics on tissues. And then finally, we have mechanical methods, and this is where we actually remove the microbe from the substance. And filtration is the only mechanical method that is effective. And in some situations, we can actually sterilize the substance when we're looking at liquids, or we can decontaminate it, and that's when we're talking about air. So we'll get more into detail into each of these methods as we go through the chapter. So some organisms are more resistant to control than others. And when we're talking about microbial control, we're primarily targeting those organisms that are responsible for causing infection or result in the spoilage of food. Typically, when we're looking at populations of microbes, we are not looking at a single species of organisms. Microbial populations are often composed of mixed organisms, and those organisms have varying resistance and harmfulness. Some are more harmful than others, some are more resistant than others. Many of these populations may include vegetative cells as well as endospores. Endospores are the most resistant microbial entity that we have to address as far as control. Prions are mentioned in the chapter, and we've discussed prions in earlier chapters. Prions are much more resistant to control methods than endospores. Unfortunately, at this point in time, we have not developed really good methods for addressing prions. These methods are constantly changing as our understanding of them improves. So when we're talking about sterilization processes, we're talking about processes that are effective against endospores. They are not necessarily effective against prions. So the goal of any sterilization process is to remove bacterial endospores. And since endospores are the most resistant structure that we're going to discuss, any process that will kill endospores will also kill any structures that are less resistant than the endospore. So this is an image from your text, and it shows sort of the relative resistance of different organisms. So you can see that enveloped viruses are not very resistant. And that has to do with the fact that they are enveloped. So they're surrounded by a phospholipid bilayer that is relatively susceptible to many detergents, basic hand soap. And then, of course, bacterial endospores and prions are at the top. They're the most resistant structures. And then you can see there's a variety of organisms and structures that fill in between. So we need to distinguish between agents and processes. So a process, we're talking about steps or methods for microbial control, those things that we've discussed already: sterilization, disinfection, decontamination or sanitization, antisepsis or degermination. These are processes. Another process is asepsis. Hopefully in the microbiology lab you have learned about aseptic techniques, and this is a process that is practiced in healthcare and a number of other laboratory environments that are practices to help reduce the contamination of materials and other substances in the lab to infectious agents. So these are all processes or methods. Then we have agents. So agents are the chemicals or other materials that we might use in those processes. So we have a number of different agents, and each agent has a varying target. So a bactericide is a chemical agent that destroys bacteria, and it is only effective against bacteria. Fungicide is a chemical agent that destroys fungi and their spores. A viricide is a chemical agent that inactivates viruses, especially those that are found on tissues. A sporicide is a chemical agent that destroys bacterial endospores. A germicide or a microbicide is a general term for any chemical agent that kills microorganisms. And this is how we're going to refer to most agents, as germicides or microbicides. An antiseptic is a chemical agent that's applied directly to body surfaces, wounds, and incisions to destroy or inhibit pathogens. Okay, so these are things like alcohol, hydrogen peroxide. And then bacteriostatics are chemical agents that prevent bacterial growth on tissues or objects. So the root static means to stop or stationary. So bacteriostatics don't actually kill any organisms, they just prevent additional growth. Okay, so they halt growth. All right, so when we're considering microbial control, there are a number of things that we need to think about, and what it is that that needs to happen. What control method are we going to use based on what needs to be addressed? Is it inanimate? Is it tissue? So Critical medical devices must be sterilized. So these are any devices that are going to come into contact with sterile tissues. So these are invasive medical devices. They must be sterilized. Semi-critical devices require high-level disinfection, not necessarily sterilization, but these are structures that are going to come into contact with mucous membranes. So things like respiratory devices. Non-critical devices require low-level disinfection. These are structures that do not touch the patient or may only come in contact with the skin, and it's intact skin. We're not talking about open wounds. And some considerations to make when we're looking at microbial control is the cost of the control method. How effective is it on that particular material or device? And then how will it be disposed of? So not all of these methods result in microbial death, as I mentioned before. Some of them just prevent additional growth. When we're talking about microbial death, we mean the permanent loss of reproductive capability. So even when the organism is in optimum growth conditions, the use of that particular method or process will result in the loss of that organism being able to reproduce. Death of an entire population is not instantaneous. Okay, it takes time, and that time depends on the process and the agent, the concentration, the amount of time that the population is exposed. It also depends on the threshold or the level of resistance of the organism. Death will proceed in a logarithmic manner, so it will start slowly, and then as the organisms or the population is exposed to that over a greater amount of time, more and more microbes will die, and that curve will increase logarithmically. Active cells tend to die more quickly, and we talked about this in chapter 6 when we talked about microbial growth, that organisms that are in that exponential growth or log phase are actively reproducing, and those organisms tend to be more susceptible to antimicrobial agents. Sterilization is when survival of any cells is highly unlikely. So when a material has been sterilized, based on the length of time, the temperature, the concentration of the agent, it is very unlikely that any microbes survived. There are a number of factors that can affect the death rate, or how long it takes to kill off a population, to sterilize or to reach disinfection. So the number of microbes has a big effect. If you're dealing with a few or many, the length of time that it's going to take to kill those microbes is going to vary significantly. The nature of the microorganisms in the population. If your population is composed of fairly susceptible organisms, then it's not going to take as long as if the population contains a large number of more resistant organisms. The temperature and pH of the environment are going to affect the death rate, because we learned again in chapter 6, temperature and pH have a huge effect on microbial growth. So that's going to affect death rate as well. The concentration we're talking about the dose and the intensity of the chemical agent that's being used. And then the mode of action. So depending on the chemical agent that we choose, that agent is going to target different aspects of that microorganism. So some agents target different things, and depending on the target, it could take less time or longer time to kill those organisms. And then the presence of any interfering substances, organic matter, solvents, or other inhibitors. So saliva, blood, feces, these can all inhibit the effectiveness of some chemical agents. So the relative cleanliness of the site that's being treated can also have an effect on the death rate. So antimicrobial agents, as I mentioned before, have a range of targets when treating microbes. The least selective agents tend to be effective against the widest range of microbes. So these are things like heat and radiation. They don't target particular cellular components. They are general. They're going to target everything. The selective agents target only certain cellular components. Many of the chemicals that we use do this. Also, our antibiotics are very cell target specific. There are 4 general cell targets when we're looking at chemical control of microbes, and that is the cell wall, the cell membrane, cellular processes like protein synthesis, DNA replication, and then proteins, so the action of enzymes and other functional proteins within the cell. So if we look at each cellular target, we can see that each agent has a different effect on that particular target. And then you can also see some examples of agents that are going to act on these targets. So the cell wall, most chemical agents are either going to block cell wall synthesis or they're going to break down the cell wall. And some examples would be any chemicals, detergents like soap and alcohol are all going to affect the cell wall. The cytoplasmic membrane, the general target is to bind to the lipid bilayer of the membrane and then open that membrane so that harmful chemicals can enter the cell. And this is the action of soap detergents. Cellular synthesis, there are a number of things that can be targeted here. So in general, we're talking about protein synthesis. So this the target is typically on the ribosome. We can also target DNA replication by preventing the copying of DNA, and some agents are going to also cause mutations within the chromosome. Some examples of that are formaldehyde, radiation, and then ethylene oxide, all target cell processes. Then finally we have proteins, and the typical action of agents used that target proteins are either to denature the protein or block the active sites on enzymes. Moist heat does this because of the temperature. Remember, proteins denature outside of that optimum temperature. Also, alcohol and any of the phenols are going to do that as well. So that is the end of mini lecture 9.1. Here are a few of our checking in questions. Make sure you go through those, that you can answer them correctly, and also review the learning objectives. And here's our critical thinking question. So prior to his scheduled knee replacement surgery, you advise your patient to shower with Hibiclens the morning of the surgery and allow the solution to remain on the skin surrounding the knee for 5 minutes. What antimicrobial process are you advising your patient to implement? So think about those 4 processes that we discussed in this mini lecture, and then determine which of those processes would this activity fit in. You can find the answer to this question in the next slide. So go ahead and work through that, and then you can find the slides in our classroom shell. And hopefully this video was helpful.